Hi class, welcome to the chapter 13 lecture. This is on muscle reflexes. We'll also spend some time throughout the lecture and at the end taking a look at a few of the PhysioX activities since they demonstrate these concepts well. So these, I'll point you in that direction when we get there. So the knee-jerk reflex is one of the reflexes that we'll learn. I do believe in utilizing the same examples over and over again and developing those when appropriate, as opposed to giving you multiple different examples to learn. So please focus on the reflexes that are contained within this chapter. So we're going to go through the first three sections of chapter 13. The other two parts are important, but we'll cover those really throughout the remainder of the semester, a little bit here and there. All right, so as we've learned already, a reflex pathway begins with some sort of stimulus. There is the afferent pathway that sends the message from the stimulus into the integrating center, which is the central nervous system. So either the brain or the spine. Skeletal muscles are integrated in the spine. The integrating center processes that information and then sends out through the afferent nerve the message that leads to the response, that effector response. So movement, with the example of touching something hot with our finger, the sensory nerves in the finger pick up that pain, that heat, send it into our spinal column where it's processed, and then out comes a muscle response to retract or pull the finger away from that burner. And it happens very quickly, as you may have experienced. All right, so let's look at the difference between a monosynaptic reflex versus the polysynaptic reflex. Mono meaning one, so same pathway that we just looked at. So we have the stimulus that's picked up by a receptor. That message travels through the sensory neuron into the spinal cord. Again, it's always the spinal cord when we're talking about skeletal muscle. So that's our integrating center. And then through the somatic motor neuron, the afferent signal is sent to the muscle, the target cell, leading to that response. So that is one reflex, a single reflex, monosynaptic. In this example, we're looking at a polysynaptic response. So we've got two different synapses happening. And the pathway begins with a stimulus that is picked up by the receptor, goes in through the sensory neuron into the spinal cord. So that part is the same. But what happens in the spinal cord is that we have this interneuron, and it is able to take in the message from the sensory neuron, process it, integrate it, and then send it out through a different afferent neuron, leading to the target cell in response. So this is an example of a polysynaptic because we have two synapses in here. Both of the synapses occur in the integrating center, and it does involve a single interneuron to pass that message. So interneurons are spaced, or we saw them in the figures of chapter 12, they're spaced throughout the nervous tissue. The central nervous tissue, I should say, so that they're able to integrate the information and pass it on. All right. So our central nervous system is able to control the muscle contraction, the length of it, the strength of it, and it does this by sending action potentials. And we know that happens through the somatic motor neuron. So what we've been talking about really so far have all been excitements of excitatory stimuli, but and the neurotransmitter that is associated with skeletal muscle excitation is acetylcholine. All right, so in muscles, acetylcholine is a excitatory neurotransmitter. We will see in other muscles that's not true. So please keep in mind that I'm emphasizing that in skeletal muscle, acetylcholine is stimulatory. It excites. So that leads us to a question: how do we inhibit a motor unit from contracting? So think about one of the antagonistic muscle sets. Let's actually talk about we'll talk about the quadricep and the hamstring. Those are examples that we can look to this, and that's associated with that knee reflex that we're going to learn about. So here it is. So the knee jerk reflex. So we've got two different types of neurons in here. So we do have a polysynaptic nerve. It begins with when the healthcare provider uses that special hammer to hit a specific part of what we'll learn, a specific part of the tendon right below the knee. So that's the stimulus. And that is picked up and sent through the afferent pathway up into the spinal column, right? So it's processed in that gray matter. And then we get a message coming out, our efferent pathway coming out, that is going to target the quadriceps. So that's the pathway that we're looking at for the quadriceps, right? We're actually keeping it simple right now. We're looking at just one of the circuits involved with the knee jerk reflex. We'll add in more. So we've got the hamstring and the quadricep that are actually part of this. So we're going to continue to add on. All right. So what that means for the what that means is with our knee jerk reflex, in order for the afferent signal to result in the target, that's targeting our quadriceps. So when our quadriceps are extended, the hamstrings are inhibited. So that's not shown, that part's not shown on the figure yet. We'll get to it. But keep in mind that with our antagonistic muscle sets, our pairs, one is active and one is inhibited. And this is due to different neurotransmitters. So acetylcholine, again, is an example, is the excitatory neurotransmitter. So in order for the knee jerk reflex to happen, what type of graded potential is needed in that quadricep? So let's run through our examples. So we've got excitatory signals that maintain a resting membrane potential. Well, let's think about that. If we maintain a resting membrane potential, is anything happening? Haven't we learned that it's a change in the resting membrane potential that is associated with the action potentials? So what about the inhibitory signals that maintain the respiratory membrane? Same principle. The inhibitory signals hyperpolarize, or the excitatory signals depolarize the somatic motor neuron. So D would be the correct answer here. So let's dig into that graded potential and using that example. So again, we've looked at the excitatory. The inhibitory pathway is associated with a different neurotransmitter called GABA. And this is showing it between the quadriceps and the triceps. So another set of antagonistic muscles. So what happens is one muscle is inhibited by GABA, while the excitatory pathway is triggered by the release of acetylcholine in these antagonistic muscle groups. So think about the location of what's happening where, right? It's got to happen in that transition that is happening in that central nervous system. Okay, So I'd encourage you to label the different parts of the diagram, apply the whole reflex pathway that we've been learning about, from the stimulus, afferent, integrating, what's happening in that integrating center, the efferent pathway out to the target response. Here, the example is trying to hold a load, and the opposing force has to be equal or greater than the force of this load. Okay, so now let's look, we'll go back to the knee-jerk reflex. It's getting more complicated because now we're showing you both efferent pathways, the pathway associated with the hamstring as well as the pathway associated with the quadricep. Okay, so the stimulus is to tap a specific part of the tendon here. So we're going to come back to that in learning a separate reflex, another reflex. So the stimulus is picked up by what are called muscle spindles. So this is a special part of the skeletal muscle that, that responds by stretch. So tapping it, that specific place with that hammer, stretches that part of the tendon, and that triggers the pathway. So we have the afferent pathway going into the spinal cord where it's integrated. We have pathway 1, which is one effect, one effector response. It comes out through the somatic motor neuron 1, if you will, targeting the quadricep muscle. And that causes the quadricep to contract, and then the leg goes up. As the quadricep contracts, it causes the swing up. At the same time, what's happening is the same afferent pathway in has been passed through an interneuron. So we have synapse happening right in the integrating center. And now it's coming out on the dotted line showing the inhibition of the hamstring muscle. And it does this by releasing the neurotransmitter GABA, and the hamstring muscle has to stay relaxed at the time when the quadricep is contracted, allowing the kicking of the leg up, allowing that extension movement. So our excitatory pathway involves acetylcholine, and the inhibitory pathway involves GABA for skeletal muscle. So please know this reflex. Don't be surprised if you're asked to interpret it or even identify the main players of it or use it as an example of an antagonistic muscle group. I do, again, try to use the same examples on my exams that, that we talk about in class. Okay, so now let's get a little bit, let's fine focus. Let's look in a little bit more. We've looked at the whole reflex, at the level of the, really the organ system, the skeletal muscle system. But now we're going to zoom in a little bit more and look at the different types of stimulus or stimuli that are present in our skeletal muscle. So we have what are called proprioceptors. That's a new word for us, but ceptors really isn't. We've seen that in receptor. Okay, so these are receptors that are located in skeletal muscle, joint capsules, and ligaments. Okay, so they're located in outside of the muscle too, but they are all tied into muscle responses. Okay, proprioceptors. And these are able to pick up input and send it to the CNS. So in fact, proprioreceptors are on. They're always sending information into the CNS. And what the CNS perceives is a change in frequency is really what happens with these. So the hamstring quadricep reflex is often tested, as you know, in a general physical exam. And what it's doing is looking at muscle tone. But in order to get that response right, it's got to go through that pathway correctly. All right, so the first type of proprioceptor that we'll talk about, there's 3, are the joint receptors. And they're located, as you might guess, they're located in the joint. Specifically, they're in the ligament. And their job is to constantly feed information into the brain so that we know where we are. These are, again, always firing. It's not a matter of them being turned on or turned off. It's a matter of there being a difference in the firing frequency, the time in between the signals that is perceived by the nervous system. But the joint receptors are really important in our muscle tone, our ability to have our posture. In order to sit here in this chair, even though it might seem like I'm a little relaxed because I'm sitting, my muscles are working. They're at that minimum state of contraction to maintain muscle tone. Our second type of proprioceptors are called muscle spindles. And these are found throughout the muscle itself. Okay, so they're actually in the muscle, and they're a type of muscle fiber that has evolved. So it's a highly specialized type of fiber. And these are associated with our stretch reflex. They too are used to maintain muscle tone. Our third example here are the Golgi tendon organs. So the Golgi tendon organ, whoops, sorry about that. They are located at the intersection between the tendon and muscle. And they too work to help with our tone, but they function they really function in a protective role. So they are what send a signal back to say this is too heavy, which if happens properly would result in the person changing their action. So that's how a Golgi tendon organs works. They do work in conjunction with the muscle spindles to do this. Okay, so here's an example of the muscle spindle, and these are adapted muscle cells that are distributed all throughout them. The bottom part is the tendon, which is the attachment point between the muscle and the bone. That's what a tendon does. So let's zoom in at one of these muscle spindles, and that's what we're looking at over on the right. So they have different nerves that go into them, right? There has to be a nerve pathway associated with these muscle spindles so that they have the ability to function. So we've got what's called the gamma nerve that is associated with the muscle spindles. And these are constantly sending information into the central nervous system so that the body is able to know where it is and know when the muscle is being stretched. All right, so we've got the gamma neuron, the gamma motor neuron that comes into the muscle spindle. Okay, now we're looking at the Golgi tendon organ, and these are right at that barrier between the muscle and the tendon. Again, the tendon is the attachment point to the bone. So let's zoom in at those. So look at all the inside of this. So the blue is the sensory neuron that is able to that is associated with the muscle contracting. And what happens when they contract, see how they're organized, the two fibers are organized oppositely. So when the sensory neuron contracts, it pulls the collagen. That's what the yellow spaghetti looking part is, it's collagen. And it pulls it, and that's the stimuli of this response. So Golgi tendons aren't as sensitive as the muscle spindles are because they're located throughout the muscle. Anytime we have like a bigger sample or more of it, you're going to get higher sensitivity than a smaller sample or where there's less of it. So the job of the Golgi tendon organ is it's really to help protect the body from lifting too much of a heavy load. So signaling to the body to stop lifting a load when it's at risk of damaging the structures of our body. Okay, now a second reflex is the stretch reflex. And this is a good place to study it in conjunction with the different proprioceptors. So in this pathway, the example of the stretch reflex that we're looking at is the arm extended with weight being added onto the arm. So right now the arm is, it looks like it's pretty 90 degree. It's able to handle the load. But as more is added on, what's happening is those muscle spindles are firing into the central nervous system more. And that is what's initiating that stretch reflex. So the muscle spindles are going to fire, triggering that afferent pathway. They're firing much more frequently now that there's more load on it. And notice the direction of the arm. So now it's not quite at that 90 degree. The muscles are starting to give in response to the stretch reflex. So zooming in at the muscle spindle, let's just run through our typical pathway. So our stretch of the spindle is the stimulus. It fires more frequently. So we have an increased frequency of the afferent signal going into the spinal cord. It's always the spinal cord when we're talking about our skeletal muscles. And that will lead to the afferent pathway, which is also increased. And the effector response is to contract the muscle. That's the stretch reflex. This is under a negative feedback pathway where once we get the response, then we're done. Once we get back to the normal homeostasis, if you will, the response turns off. So the reflex is initiated by sensing the different position of the muscles in response to overstretching. So our stretch reflex does involve the stretch, the stretching of the spindle itself, but let's think about it a little bit more on maybe a bigger level of organization. It's also important in our body knowing what's happening where and using and compensating in a way that hopefully protects the body. So what I'm speaking about is the arm going down lower as the increased frequency of the afferent pathway is processed. So the frequency rate would go back to normal, right? And that turns off the pathway. So muscle spindle is an increased frequency into the spinal column, increased afferent pathway out, leading to contraction of the muscle. And then once we get back to the normal frequency of firing for the muscle spindles, then a negative feedback is sent to turn the response up. So these are the alpha motor neurons. So associated with the muscle stretch, the muscle spindles. Earlier, a few slides back, we looked at the gamma neuron. So now we've seen both the alpha and the gamma. So what the alpha does here is it's targeting these fibers in the stretch response and causing it to contract. So the alpha neurons initiate the contraction, and then our gamma neurons we saw in the knee-jerk reflex, they stimulate the contraction of the interfusial fiber. So it's a different location, different part of the muscle is what we're really talking about. We've got our extrafusal fibers versus the intra, which is within fusal fibers. Okay, so I'd encourage you to go ahead and label them. Refer back to that earlier slide, a few slides back, that has the gamma pathway label for you. All right, so that's actually the end of the slide set for this chapter, but we're not done. We're going to take a look at a couple of the PhysioX activities that are in the Mastering Online Access. So let me just get to those, please, and I'll share that with you. So these are in exercise 2, the skeletal muscle physiology section. You don't need to do anything other than watch them. This is here to help your understanding of what's happening, so you hopefully can understand the lecture content a little bit better. So I'm going to, I have to turn on and off the share or else you won't be able to see it. So for those of you that want to do this on your own, I'm in exercise 2 and I'm going to do activity 2 to begin. Let me get that up and share it with you. I am going to skip over the introductory material and really just get to the demonstration. All the PhysioX labs have a pre-lab in them. Okay, and I'm going to skip the questions too. I want to get to the experiment. Okay, so the setup is we have a muscle that is suspended. It is connected. There are probes going into it. So it's connected to a chart recorder. So essentially this is an EMG, similar to what is done in the lab experiment. So what we're going to do is change the voltage and look at the effects on the muscle. Let me see if I can get my screen, get this a little bigger. PhysioX is not the most user-friendly to demonstrate because of the way it's sized, the way they size it. Okay, so hopefully you can see everything. Okay, so right now the voltage is set to 0 and we're gonna hit stimulate to start this process. So the muscle length to begin with is 75 millimeters. And look at the line tracks across. So what we're looking at in the graph on the x-axis is time in milliseconds. On the y-axis is force. So we're looking at the strength of the contraction. Okay, let me hit next. And now we're going to record our data in the data table. So at 0 voltage, there's no change. The length is 75 millimeters. Okay. I'm going to actually see if, oh, it won't let me skip them. Okay, what do you observe in the active force display when the stimulus voltage is set to 0? Well, we didn't, right? It was a straight line across. So what's our options? We've got there was no activation of skeletal muscle fibers by the stimulus. The low stimulus voltage activates only a few. No. Once skeletal muscle is isolated from the body and mounted, it can no longer generate force. It can under artificial conditions, because that's the whole point of this. All right, so we are going to, that's the same thing. You got to follow the steps in PhysioX or it doesn't work right. Okay. Oh, it's doing the same thing again. Next. Okay, no, sorry about that. So now it's telling me to increase the voltage to 0.2. So that's what I'm going to do. Before it was 0. And now I'm going to hit stimulate and we'll see if this is enough stimulus, enough electrical stimulus. We're looking at that blue line and it just goes straight through as well. We're going to record our data. And what we're going to do next is we're going to increase the voltage that's used to stimulate the muscle so we can figure out that minimum voltage that's needed. So it tells us to increase it by 0.1 at a time. So let's do that. Now we're up to 0.3 and we're going to hit stimulate. There it goes. It didn't do anything again. Record data. I wonder. Okay. Okay. Let's hit stimulate again. We're at 0.4 now. That blue line is what we're following. And it still didn't do anything. Let's record that. Now we're going to increase the voltage to 0.5. Stimulate. We're looking at the yellow line. Nothing. Record data. Let's go to 0.6. Remember, the instructions told us to do 0.1 at a time, so I'm following the instructions of the PhysioX activity. Still nothing at 0.6. 0.7. Nada. 0.8. Oh, we got a twitch. Did you guys see the muscle actually twitched? And there is a little blip. It's hard to see on the chart recorder, but I think we're going to see something more now. Okay, we're going to go to 0.9. Watch the muscle as well as the chart recorder, please. Stimulate. Oh, there we go. Now we can see. So the more voltage, we're getting more force. Let's go to 0.1. Stimulate. Record. 1.1. So it produces a nice pattern where does look like there's a relationship between the increase in voltage and the increase in the force. Okay. So what is the lowest stimulus voltage that induces active force in the skeletal muscle? What is that called? That is the threshold. Okay. Next, let's continue. Actually, that's the end of this experiment. We're going to look at another condition. So let's clear these and let's see. Next, okay. Increase the voltage stimulator to 1 volt and hit stimulate. We already went there. We're going to record that data. Next. Now it's telling us to increase it by 0.5. So I'm going to go to 1.5. Stimulate. Now we're getting a bigger response. Record. We're going to do this until we get to 10 is what the instructions say. So we're going to go to 2, stimulate. Sorry. Record. 2.5, stimulate. Record. It's going to take a while to go to 10. There's 3. So we've got a gradual increase. I don't know if it's proportional without looking with a statistic, but it looks like it is. You can't just always assume those things by looking at data, right? Statistics actually are measurements on a data set. Record. Okay, now we're going to go to 4. Nope, I was trying to find a quicker way. There isn't. So at some point, are we going to find a voltage that does not have an effect is what we're going to be able to determine. So we are at we're halfway done with this part of the experiment. But notice the increases aren't as big in between anymore, right? The distance in between the lines, it's not as big as it was in the beginning. We're at 5.5. Now we're going to 6. Oops, I went past. 6.5. 7. 7.5. 8. Oops, now we're going to 8. Thanks. There we go. Yeah, those lines are starting to touch each other. We're almost done with this one. Oops, 8.5. Got 3 more tests. 9 volts. We're right on top now. 9.5. Notice the lines are overlaid now. There's not a change. So what could be causing this? There we go, record. Okay, let's see what's next in this experiment. We are going to enter the maximum voltage for this experiment in the field below. So our maximum voltage was 10, right? We went to 10. That seems too easy. Okay, well, the point is that as we increase voltage, we got an increase in response shown by an increase in the force, that strength of the contraction. However, it got to a point beginning, what, about 8.5, I think it was, 8.5 volts, where there no longer is an increase because we're at the maximum for this. All right, so that is it on this particular experiment, but we are going to do another PhysioX. So let me get set up for that, please. Okay, so now we're going to look at activity 4 of, actually, no, let's go ahead and do activity 3 of exercise 2. So we're looking at stimulus frequency, the effect of stimulus frequency in this experiment. Let me share the screen with you. Okay, so very similar setup. It is beginning with a voltage of 8.5 volts and the muscle length is 75. So we're going to hit stimulus, single stimulus, and we're going to get a response, right? We've already seen that this is enough to lead to a contraction. Record data. Let's see, and now where does it tell us to go? Select single stimulus and allow the trace to rise and fall immediately after the, oh goodness. All right, I've got to start that part over because I didn't read it quick enough. So what it wants me to do is hit single stimulus right after it falls to baseline. So that would be about 120 milliseconds. So I'm going to do this twice again because I need to do the first part so I can hit the second part. So here we go on the single stimulus. I'm not going to record it because it's the exact same thing. And now there we go. Didn't do anything for us. Okay, we did record. Okay. We're going to hit next. And now, well, we should have observed an increase, but we didn't. Let's start over. Let's clear those traces. Okay, so I'm going to start over now. And as soon as it falls down, there, now I've got to, I'm going to do it one more time. Okay, as soon as it gets to baseline, I'm going to hit the single stimulus again. There we go. So notice the second twitch was bigger. Sorry about that. I didn't do the timing right. But because that second stimulus came in before, before the end of the cycle here, we got a bigger response. Let's record our data. and next. All right, so now we're going to start to look at summation, which is something we talked about in chapter 12. We've got different types of summation. So let's go ahead and take a look at what that is. Let's see, we're going to select single stimulus and watch the rise and fall. These are the same instructions we just did. Let's move forward. Okay, so now we're going to stimulate the muscle at a higher frequency by hitting that single stimulus 4 times in rapid succession. So I'm going to clear the tracings and I'm going to go ahead and do that. So 1, 2, 3, 4. So notice it increased it up to a much higher force than we've seen. Clear tracings. Let's get our next conditions. Okay. Now we're going to increase to a voltage of 10 from 8.5. And let's see, we're going to hit single stimulus 4 times. And so we're at a higher voltage and we're going to hit it 4 times. Ready? Let's go. 1, 2, 3, 4. Pretty much the same pattern we saw. Record. But our total force is different, right? Our total force between, oops, let me get to it. It's actually the same in the last 4. So we went from 8.5 to 10, but not much of a difference. Let's get set up for our next one. So we're going to take the voltage back down to 8.5. Oops, sorry about that. After setting the voltage, select single stimulus as many times as you can in rapid succession. Yeah, and we've got to do this until it happens, it says. So until we get an active force of 5.2. So we've got to get above a force of 5.2. So I'm going to hit it as fast as I can, trying to get it up there. Here we go. Oh, there we go. Record. Let's do that again. I went a little bit higher. Record. Okay. Okay, so our active force was higher with the increased frequency. We went from 4.65 to a 5.6 something. Okay, clear tracings, and let's see what's next. Okay, we're done with this one. Let me get to the next one. Just give me a moment to get everything ready, please. Okay, so now we're going to look at tetanus. So we're going to look at tetanus in an isolated muscle, and then we'll do another exercise, another activity where we look at fatigue. So we are doing exercise 2, activity 4. Let me just getting set up. All right, so we're at 8.5 and the stimuli per second now is set at 50, and we've been looking at single stimuli. Now we're looking at multiple stimuli. Okay, so this is going to show us unfused setness. So what we're going to do is make sure. Okay, I just want to make sure I know where everything is. Here we go. Oops, there we go. All right, it's still going, even though I hit stop. So let's go ahead and do that again. Why are we still going? It's not done. All right, I think we can see what happened though with the frequent multiple stimuli. Look how high our force began to begin with. Okay, so now we're going to look. So this is unfused because we still have some variance. The muscle is not let's see, the frequency of the stimuli are far enough apart that it's unfused tetanus. We're going to now look at fused tetanus. Let's get rid of this. Okay, so let's increase the stimuli by second to 130. I don't think it lets me type it in there. No, it does not. Okay. Multiple stimuli, and then it tells us to stop it once it starts to wrap around the screen. Okay, I stopped it. Takes a little bit of time. So it's more flat in the fused tetanus. Let's record that. So the force is higher, right? We have more stimuli per second, 50 versus 130. It's the same voltage. All that we've changed is the frequency. So that if the frequency is high enough, then that can lead to fused tetanus. All right, let's go forward. Clear tracings, yes. We're going to take this up to 140 now, 140 stimuli by second, and do the same thing. All right, record. Okay, so now we're going to look at the effect of incremental increases in the number of stimuli per second above 140 stimuli per second. So it says to increase the stimuli per second setting by 2. So we're going to go to 142. Whoops, it does it in 2. Okay, and then do the same thing again. Here we go. Record. Clear tracings, and now we're going to go to 144. It's getting up there quicker. 146. Oops, I should have cleared the tracings. But it's a different color. It's actually right on top of it. I'm glad I didn't record it. That was interesting to see. Let's clear the tracings, and we're going to do 148. So it's getting very quickly up to the top, and there's no variance now. Fused tetanus. And now we're going to go to 150. Clear tracings. Multiple stimuli. Here we go. Record. So let's look at that data. So going from 140, so we're at 5.9 at 140, the force. At 142, it's the same thing, 5.92. 144, 5.94. 146, 5.95. It's not changing. Okay, so we've hit, because we've hit fused tetanus. If you just keep going higher, right, you're already above that level that's needed to hit tetanus, so it's going to do the same thing. You can't go any further. There's nowhere to progress to past fused tetanus. All righty. So that is that one, and we've got one more activity we're going to do. We're now going to do activity 5 of exercise 2, and this is looking at muscle fatigue. Here we go. So we're at 8.5 volts and then 120 stimuli per second. We're looking at multiple stimuli. So it asks us to select multiple stimuli, observe the muscle force tracing, and stop after the force falls below 0. So once it gets back to baseline, I'm going to stop it. There we go. Record data. Clear tracings. And there's no next button. Why do the stimulated muscle force begin to decrease over time? It really didn't give us much of an experiment to look at here, despite the maintained stimuli. So I'm going to actually go back and do, nope, that's all it's going to let me do. That's weird. I was trying to repeat it again for you, but it doesn't give me the option to do that. So that does not seem right. Let me go ahead and set that up from the beginning again. Sorry, it's a good activity to do, but I'm not quite sure why. I must have skipped a step. Okay, so let's start over. Okay, so we're going to do that again. Stop it when it gets to 0. There we go. Record data. And now it says clear tracings. Okay. Why does the stimulated force begin to decrease over time despite the maintained stimuli, right? Because we've held the stimuli constant at 120 stimuli per second. That part hasn't changed. So but we did see, right, the curve came back down. So there's definitely a change in force. The force got back down to 0. So what do you think could be happening? Is there, are we using an alternate source of energy to deal with fatigue? Would that be a buildup of ADP and inorganic phosphate? So actually that would indicate that we've got phosphocreatine being harnessed out of it. Could it have to do with the intracellular calcium movement not being impaired? In order to move into the next cycle, the sarcoplasmic reticulum needs to regain all of its calcium so that it can be or regain enough calcium so that it can be released with enough gradient into the synaptic cleft. Okay, here we go. Now we get to change it. No, those are the same instructions. I'm going to do it one more time, since that's what it says to do. Stop, record. Clear tracings. That's the same question. Yep. I'm not sure why it seems to be stuck. Let's see, I'm just kind of guessing. Yeah, that's what we put. We did put more than one answer the first time, but I don't know why it didn't do it. Anyway, if the stimulator is briefly turned off for defined periods of time, what will happen to the length of time that the muscle is able to sustain maximum developed tension while the stimulator is turned on? So we're pausing the frequency, and it's asking us what will happen to the length of time of sustaining the tension. Well, if it's paused, that could provide the opportunity to come out of tension, right? If it drops back down. So we've got the length of the rest period will not substantially impact the length of time for sustained muscle tension. The length will proportionally increase it, or the length of the rest period will proportionally decrease the length of time for sustained muscle tension. It's going to decrease it, right? Because if you drop out of tetanus, you're out of tetanus. All right. So now we're going to look at our different conditions finally. So we were going to look at multiple stimuli. After the force falls to 0, we hit stop, and then we wait 10 seconds and then hit multiple stimuli again, and then stop it, and then wait 20 seconds. Okay, here we go. So the first wait is 10 seconds. The second wait is 20. Ten seconds, start. Oh, I did that. Oh, it doesn't for me. Even better. Stop. Cool. Okay. So the shape of the curve looks pretty similar, right? The total force is the same. The distance in between is different, but we controlled that. Now I'm stuck again. There we go. Record data. Why did the length of the intervening rest period affect the length of time the muscle can maintain tension? So we've got, let's look at the data. So our rest period of 0, our sustained force, our active force is 5.86 with a sustained force of 0.8. With a 12-second rest period, it's basically the same active force, but when we have a longer rest period, we have a longer sustained maximum force. So the muscle's adapting. It could be firing a different motor unit to prevent fatigue. There could be a quick recovery of the calcium so that it can be recovered and go into the next cycle. Let's see, would intracellular concentrations of lactic acid increase in the rest? Okay. Would intracellular concentrations of acetylcholine increase? So is it still, in order for acetylcholine to increase, it has to continuously be fired. Intracellular concentrations of ADP and inorganic phosphate declined, or the calcium levels in the sarcoplasmic reticulum declined during the rest period. What do you think? Let's see. So we go from 0.08 to 0.02 to 0.06. I'm trying to really appreciate a trend there. There should be, every time it's being stimulated, it's a new cycle, right? So our calcium levels are able to get back. If there's a longer rest period, it would make sense that that would give the cell time to recover its calcium levels. But that's saying that they declined in the sarcoplasmic reticulum. Let's just see what they say. Yeah, so our ADP, we're running out of nutrient, out of energy with this process. And the longer the period of time is, there's more energy needed, right? So cool, that's it for this one. Okay. All right, so hopefully those help to demonstrate some of the properties of skeletal muscle. And those of you who don't have PhysioX, you got to see it. There actually is one more I'm going to show you, and this is on looking at load on the length, or the length and the tension relationship in skeletal muscle. That's a good way to put it. So let me just get to the experiment part of it. All right, here we go. Let me share this with you. Last share, I promise. I just want to make sure that people that don't have access to PhysioX can see this, because it is good information. So we're at 8.5 and we're at 75 millimeters in length. So we're going to hit stimulate and watch what happens. So we're looking at the So we go up to about 1.8 and then we drop back down. So over on the right side in red is the active force, green is the passive force, and then the yellow is the total force. So we've got an increase in the active force, right? That would make sense. Passive force wouldn't if we're trying to stimulate. Okay, let's see the next instructions. It said we should see a muscle twitch tracing on the left. We did. And we have 3 data points, but 2 of them are overlaid. Okay, let's see. Did I record? No. Okay, as the resting length of the muscle is changed, what will happen to the amount of total force the muscle generates during the stimulated twitch as the resting length of the muscle is changed? So that again is an increase in the time. That should, but this is resting length. We're not holding a contraction. What did we see it do? Let's go ahead and stimulate that again. Let's look at the length of the muscle. Does it change? It's still the same. We didn't see a change. So an increase in muscle length will decrease the force. An increase in muscle length will increase force. A decrease in muscle length. Total force can increase or decrease depending. Let's try that one. Good. All right, so now what we're going to do is we're going to shorten the muscle and then look at the results. So it tells us to shorten the muscle by 5 millimeters, stimulate, record, and we're going to do it until we get to a muscle length of 50. So we're starting with 75. So we're going to go down to 70. Clear tracings. Let's stimulate. And record. All right, so looking at the graphs, we've got our active force, our passive force, and then our total force. They're slightly different between the two. Okay, let's clear the tracings, and now we're going to go to 65. We're going to stimulate. Record. So look at our active force. It's decreasing as the muscle length decreases. Our passive force is still 0 because we're active. So let's plot that data. So now we've got a bigger drop, right? The other two were closer nestled together. Clear tracings. We're going to go to 60. Notice the muscle is getting shorter. Stimulate. Record data, and now our new data point is here. Big drop off. Clear tracings. We're going to go to 55. We're almost done. Big drop. And our last setting is at 50. We're going to stimulate. Whoa. Look how much lower it is. I left the previous tracing on it, but it's helpful to see it. Okay. Record data, plot data. Okay, so now we're all the way down here. So the muscle's too short to work properly now, is the easy interpretation of this. Let's see. Now we're going to lengthen the muscle to 80. We're going to make it longer. We've been looking at shorter. And it says to stimulate and to deliver the electrical stimulus to the muscle and note the values of the total active and passive forces compared to what we saw when the muscle was 75. Was that a higher force? Let's look. Oh, yes, much higher force. So we're back up here. Okay, so now we're gonna vary the length of the muscle. So we're gonna lengthen by 10 millimeters at a time until we get to 100. So we're gonna go to 85. Stimulate. I'll look at the graph after we go through them all. Record. We're going to go to 90. Stimulate. Ooh, it's dropping off a little bit. Look at that. And we're not going down to baseline. Very interesting. I hope it gives us a different color. Nope. So we're still lower, but look what's happening now. The time to attempt to get to baseline is even longer, and it fails to do that. Let's go to 100. So we're looking at this blue line way up high now. Goodness, a higher force. Oh, wow, our force is staying the same. That's interesting. What does that mean if a force stays the same? Is that tetanus? Let's look at the plot. Okay, so we added a bunch of points up at the top, but look how pretty this is. Look at our active force and our passive force. So now we're starting to see a passive force with those latter ones because we don't get back down to baseline. So there is a passive force. So as the muscle lengthened beyond a certain point, it changed the dynamics of the active versus the passive force. Let's see what's next. That's it. Okay. All right, class, that's it for chapter 13. I hope you're having a great day or night, wherever, whenever it is you see this. Take care. Please feel free to reach out by email at any time. Bye-bye.